Today I want to explain the molecular mechanisms which underlie cell motility. And in particular, I want to focus on this lamellipodial region of crawling cells, which is really rich in actin filaments, which we introduced in the last class, and also bring in the idea of myosin and try to explain how myosin works. And as just to kind of start off, here's the classic picture of a crawling cell. So once again, this is a leukocyte or a white blood cell that's migrating around. This is the tail of the cell, sometimes called the uropod. This is the lamellipodium, and there's a little bacterium here. And what we want to do is understand the molecular mechanisms which allow cells to generate forces and crawl around in the environment. As just a little bit of review, in the last two classes, we talked about some of the proteins that are important for modulating or regulating actin assembly. As just kind of a brief recap, when G-actin is bound with ATP, it preferentially adds onto the plus end of actin filaments. Over time, that ATP gets hydrolyzed to ADP plus phosphate, and then that phosphate is eventually released, and we have ADP actin at the end here. There's a protein called cofilin that comes in and it cleaves the actin filaments and breaks them into small little fragments with lots of myosins from which actin can depolymerize. This G-actin then can interact with profilin. Profilin promotes actin filament assembly. In part, what it does is it binds to the G-actin and it opens it up so that that ADP can be released and then replaced with ATP. There's another protein, thymosin beta-4, acts as a bit of a buffer. We're not really going to discuss it today, but you have this general cycle where ATP G-actin is created, added onto the filament, and then it's processed and then the cycle continues. Starting from a very sort of elementary or reductionist understanding of actin dynamics, we then build up what is called the dendritic nucleation model. And actually, I'll start off and ask, does anybody know why the dendrites of neurons are called dendrites?
Is it like roots?
Roots? Actually, that's a good question. So what does dendrite mean? It means branched or tree-like. So yeah, so somebody looked at the dendrites, the extensions of neurons, and they said, wow, it looks like trees extending out. We'll call them dendrites. So this is also called a dendritic nucleation model. It doesn't have anything at all to do with dendrites and neurons. What it's more referring to is that when you have these branched structures, it looks like a tree. So in the last class, we introduced this dendritic nucleation model, and this starts to put the biochemistry that we initially discussed into the context of a whole cell. And once again, we're looking at the lamellipodial region of the cell. We have the plasma membrane here up at the top. There's going to be receptors that can sense extracellular stimuli. And what they're going to do is they're going to lead to the production of PIP2. So PIP2 is a molecule associated with the membranes that is very commonly associated with high rates of actin filament assembly. They're also going to activate GTPases. In this particular case, WASP is going to be activated, and WASP is an activator of ARP23. So ARP23 is actin-related protein, subunit 2 and subunit 3 that come together. And what they do is they bind to the sides of these actin filaments, and they look like the plus end of an actin filament. And from those sort of pseudo plus ends, these new branches can assemble, and this leads to this dendritic organization of actin filaments at the leading edge of the cells. As before, ATP associated with G-actin binds to the plus ends of these actin filaments, and over time it gets converted to G-actin with ADP. This gets broken down, and then we can see these subunits that are being regenerated by prophylin, and then they can eventually come back in here and serve as sources of material for new assembly. And I'll just pause for a second and ask if there's any questions on this model. Okay, all right. So what we're going to try to do is understand how this model comes to motion, comes to life, and helps cells move. And just to sort of start off, this is the growth cone of an ecclesia neuron, and it has been labeled up with a marker for actin filaments. The C domain of the growth cone, the central domain of the growth cone is over here. There's going to be lots of microtubules and organelles that are found in the C domain. The P domain, or the peripheral domain, is out here. This corresponds to the lamellipodium. And I guess and then the plasma membrane is going to be here. And what should be fairly obvious here, I hope that you can see, is that there's this motion that's showing up where things are being moved back to this region here. And this region here is very special. In growth cones, it's called the T zone or the transition zone. In migrating cells, it's often called the convergence zone. And this is where material is flowing into and it's being recycled. This is where the forces are really sort of being generated that, well, is helping to drive this motion. And we'll try to do this. So actin filaments move rearwards to the leading edge of the cell. And some questions are, what's driving this motion? And as just to think about it, let's say that you were sitting here looking at these cells and you wanted to look and ask yourself, what's driving this motion? What are some of the things that you think that you might want to look at, especially given the knowledge that you have about actin dynamics and stuff? So yeah, go ahead.
You might want to find out the distribution of motor proteins at that region of the cell.
So the first thing that you can do is a little bit of immunocytochemistry or fluorescent tagging of proteins to try to find the localization of proteins in the cell. And when people have done that, one of the things that they do find is that, in fact, myosin does tend to be concentrated here at that convergence zone. So that would be kind of an observational sort of thing. So let's say that you have that knowledge in hand, then what are some of the things that you might do? So yeah, myosin is enriched here. What next?
Is this a migrating cell or what cell is it?
In this particular case, the growth cone is not extending, so these neurons were plated on polylysine, and for whatever reason, when ecclesia neurons are grown on polylysine, they don't elongate very much. They do, but it's at an extremely slow rate. So over the course that we're looking at, there's no cellular motion. The position of that convergence zone is going to stay put.
Okay. Maybe the velocity of fluid flow around this region.
The velocity of fluid flow, so are you talking about in the extracellular media, the water flow, or?
The intracellular media, like cytoskeletal flow.
Yes, yes. And in fact, that's what we're looking at here. So these are actin filaments that have been fluorescently labeled. And actually, I should maybe even explain that a little bit. So this is using fluorescent speckle microscopy, a very low concentration of these. And actually, yeah, if we look at this, you can sort of see little speckles show up in front. What's happening there is the dye is binding to the actin filaments, and then it's being sort of transported back. Okay, so actually, we'll just sort of go ahead and move forward, but we're definitely interested in myosin. In terms of actin dynamics, so if you blocked actin dynamics, what do you think might happen? If you blocked actin, is something here? Yeah.
We would stop this, like we would stop seeing it consistently.
Yeah, you would You would expect that there's going to be a blockage of flow here and stuff like that. And we'll go over some more details here in just a little bit. So, okay. So, when one wants to look at how cells work, it's important to have molecular tools to manipulate things. And a classic sort of approach is to use pharmacological reagents that inhibit the activity of different proteins. And these are incredibly useful tools, and they're used throughout science just to understand how cells works. In the terms of actin, there's 3 drugs or toxins that are used all the time. So the first is cytoskeletal, and the way that cytoskeletal works is that it binds to the plus end of an actin filament and it blocks it. And when cytoskeletal is bound to the plus end of an actin filament, it blocks assembly. Another protein that is used very commonly is latrunculin, and it binds to G-actin and it blocks polymerization. And I'll just ask, what is another protein that actually has a similar function but a physiological role inside of cells that we just mentioned? Yeah, so thymosin beta 4 kind of acts like latrunculin. So it sequesters that G-actin away, and it essentially lowers the concentration of G-actin, and then that's when it causes actin disassembly here. And then once again, so cytoskeletal is going to be binding right here at the plus end. And then finally, like, this image that I'm showing you here was created by using what is called rhodamine phalloidin. And phalloidin is a toxin that's found in some types of mushrooms. And what it does is it binds the actin filaments themselves, and it blocks depolymerization. And just to sort of like talk about that, so here we have an actin filament. So phalloidin is going to bind to the filament itself, and for reason, well, it seems to inhibit the depolymerization, especially at high concentrations. As to the mechanism, I'm not exactly sure what the details are. Yes, go ahead.
Would that be the reason then that we see in the fluorescent microscopy video that we were showing? Like, is that the reason that we're able to see actual clear lines of actin filament?
Yeah, yes, exactly. So one of the reasons, the question is phalloidin binding to actin filaments the reason that we can see these nice clear lines? And indeed it is. If you recall back in some of the earlier electron micrographs I had shown, the filopodia are just tight bundles of actin filaments. And one of the reasons that these lines are here is that there's a really high density of actin filaments. The dye that people now use is suractin, and it uses sort of a related mechanism to bind to the actin filaments and stuff like that. So, okay. All right, so these are 3 tools that are really useful. So we can block the plus ends of actin filaments, we can bind up G-actin, and we can also inhibit depolymerization. So, all right. So this is a movie where cytoskeletal was added to growth cones, and it's actually one of the more famous movies in like cellular, like cell biology of neurons and stuff like that. And just to kind of explain things, so once again we have the central domain of the growth cone here. This is going to be the leading edge of the cell. These long finger-like projections, these are the filopodia. And you can see the ribs of those filopodia sort of extend back here. That's usually what's really bright. In this particular experiment, bees were added to the media, and what they do is they sort of randomly bind to the growth cone, and by tracking the bees, we can track the flow of material across the growth cone. And in this particular case, there's going to be a pre-drug sort of situation where we're going to watch the movie, and then cytoskeletal is going to be added, and then we're going to go through what's happening here. So, so there's a video over there. Stuff is, well, actually, I'll play it through. and then the cyto D is washed out. So let's play through that again. So pre-drug, in this first half of the movie, this is the leading edge. What's happening with the beats?
Moving towards the center.
They're moving, yes, so they're going to be moving towards this convergence zone. And then cytokinin is going to be added, and then let's watch what happens with that. Okay, so cytokinin is added, and do you see a change in sort of like the ketamine out here? And so you sort of see this lumpy stuff, and then there's kind of a clearer thing, a region here, and then we still have the leading edge right here. So let's talk about how that all sort of works here. So we've got that, cyto D is added, all right, and then it's washed out. And I'll just sort of walk you through some questions here. So immediately after cytokinin application, what's happening here? So this is a slow down a little bit. So cyto D is going to get added. There it is. And then the question is, what's happening here? So we really want to see that. So what is happening right after cytoskeletal application here? Yeah, go ahead.
Our filaments stop binding at the leading edge, and all the filaments are seen to just kind of converge altogether.
So there's, yeah, so the filaments stuff out here seem to disappear. So any other thoughts or things? So yes. Yeah, go ahead.
Yeah, I was gonna say like the filopodia extensions, like they don't move anymore towards the convergence zone. It's just at the perimeter of the cell.
Yes, so they are just out there, and arguably what's happening is this is just some plasma membrane that has receptors on it that are allowing it to stick to the substrate here. So you can think of this as almost an empty sock. Okay, all right. This flow continues, and there's this gap. And I will say, so when actin filaments assemble, one thing that they can do is they can push things forward. And this is what some people call a molecular ratchet. And kind of the idea is that the plasma membrane is flopping sort of back and forth, and then by chance, new G-actin can form a filament, and essentially the cells are making a little stick. So as the membrane fluctuates back and forth, little gaps open up, those gaps get filled, and then that pushes the membrane forward. So through actin filament assembly, it can push plasma membranes forward. And one question is actin assembly driving this retrograde flow? And one thing to ask when you look at this movie, does this flow continue immediately after the application of cyto D? So do the beads continue moving backwards after cyto D is added? After this gap forms, do the beads start moving backwards? And I'll play it one more sort of time. You see that? Okay. And yeah, so so we still see this motion after actin filament assembly has been driven. And I'll just ask, does this imply that actin filament assembly is driving this retrograde motion? So you still see this retrograde flow, but actin filament assembly has been stopped. Does this mean that the filament assembly is driving the flow, or could there be something else going on here? Exactly, yeah. So the fact that this flow continues when the actin filaments are away from the leading edge suggests that there's something else going on here. Okay. It should be absolutely clear, yeah, actin filament polymerization is happening at the plasma membrane. And I will say there's a lot of experiments that show it can drive this retrograde flow or make a contribution. But there's something else besides actin polymerization that's happening here. So a second sort of question is what happens when cytokeratin is being removed? So during the recovery phase, and I'll just move that here. Oops. All right. So we have cyto D, and now it's going to get washed out. And then let's watch what happens. So, and I can play that in one more time. So I want to focus on right in here. All right. So the drug that's blocking the plus ends of actin filaments is removed, and then what's happening right after the removal? I'll just give somebody else a chance. Does anybody else want to jump in here?
It's like actin starts to bind where it wasn't before, it was locked.
So actin filament assembly starts to occur, yes, and
That was what I was going to say. And when it binds like that, it's still having the retrograde action, so it's just binding and then still converging.
exactly. So there's another thing. You do see this move backwards, and I'll actually, point out maybe something a little bit subtle. So right after this happens, there's going to be minus ends of actin filaments that are going to be moving backwards here. So those are minus ends of actin filaments that are being pushed backwards, and the plus ends are here. So this actin assembly can actually contribute to this retrograde flow, as I'll show later on and stuff like that. But the short of it is, it's a really complex process. There's a lot of things that are going on here. So a lot of that sort of introduction, well, this really is a very famous experiment from Paul Forscher's lab, where Paul Forscher was one of the main scientists that had worked on this problem. And it really set up the idea that actin dynamics are important for growth cones. Another molecule that is important for creating motion in cells is myosin 2. Non-muscle myosin 2 is found in all of our cells. It's a very ancient molecule. Amoeba have it, we have it. Skeletal muscle is a more recent sort of innovation. So if you think about it, things with skeletal muscles evolved much more recently than things with cells. So, but in any case, these two molecules are related. So both skeletal myosin 2 and non-muscle myosin 2 are very similar in a lot of their sort of properties. And right now, we're looking at sort of generic myosin 2 in motion. Here is an actin filament. This is the plus end of the actin filament. This is the myosin. And then this is going to be called what's a thick fiber. So it's a bundle of non-muscle myosin 2 or skeletal muscle myosin 2. And this is the myosin head, and there's the neck. And we'll put it in motion and talk about how myosin 2 works. So myosin is going to bind to the actin filament, ATP is going to get hydrolyzed, ADP is going to be released. So it's taking a step, and when I'm going to show a full molecule of myosin, this is just one head group. You're going to see that there's a lot going on. But very much, the myosin is grabbing onto the actin filament, it's contracting, and it's pulling things back. It turns out there's a lot of different types of myosin that serve different functions in cells. I think that there's somewhere over 15 different subtypes of myosin. The numbers seem to always go up over time. In any case, we just sort of talked about myosin 2, and here would be a bundle of myosin or forming a thick filament, and these are the active filaments. Another important myosin is myosin 1. It is involved in vesicular sort of activity, endocytosis in particular. And then for the point of really trying to understand the biophysics or the physics of how myosin works, we're going to focus in on myosin 5. Myosin 2 is usually used to generate large forces. Myosin 5 more often than not is used to transport vesicles or to cause vesicles to dock in certain locations. And the different proteins, they have different functions, and they also have different structures. And if we look here, and I'll just sort of point this out. So here's myosin 2, and here's myosin 5. These are the regions that are going to bind to the ATP. And you see that there's a big difference in the neck domain here. So this will be important, and we'll come back to that. All right. So in the convergence zones of cells, in the T-zone of growth cones, and actually throughout cells, myosin 2 forms these filaments. And the tails all sort of line up, and they form this nice sort of bundle. And then the heads stick out, and they're ready to grab onto actin filaments. But these myosin 2 filaments are a functional unit. In terms of the domains, I sort of had introduced this. Myosin 2 as a molecule consists of 6 polypeptides. There's these 2 heavy chains that we had just sort of talked about, and then there's 4 light chains. So there's a regulatory light chain, and there are certain drugs, that can target the regulatory light chain. And then there's also essential light chains. And one of the major things that these light chains do is they're binding this neck domain, and they help stabilize it. So for motors to generate forces, things need to be stabilized, and these light chains are important for that. And if you were to go back in time, or actually, and we wanted to figure out which part of this protein is the motor, so we have this protein here, and we want to figure out where is the motor. What are some approaches that we can do to answer that? Yeah, go for it.
Well, you just mentioned that we can have specific drugs that can target specific parts of the myosin, so we can have different inhibitors for the neck or the head or the tail, and see in different experiments which one changes the motor function of the myosin most.
That is a very nice thing to do. It would be a bit complicated. You would need to know where the drugs are binding to the proteins, and very often when you first are looking at a protein, that's hard information to come by. So what would be maybe yeah, go ahead.
You can stain parts of the protein and see which side is moving much more rapidly in the cell.
That is very cool. So you can tag various parts of this protein and look at what's moving. And actually, we'll go through some experiments that do just that, and we'll talk about that a little bit later on. That is, once again, an incredibly hard and sophisticated thing. If we wanted to do a really simple assay for figuring out what part is the motor, what might we do? And, yeah. So we have all of the molecular genetic tools in the world. We can engineer constructs. We can make drugs that cut the protein into chunks. Okay. So, yeah.
I don't know if this is like recent or old. Like, what about gene editing?
Yeah, so a classic way is essentially you could use gene editing to remove different parts of the protein and then look at where function is lost or preserved, and then you can start to assign different domains. Another thing that's very commonly done is to actually express a short fragment of the protein, isolate it biochemically, and then add it to solution and see if it recapitulates the activity of the whole protein. And then that's good evidence that that's the functional thing. In the days of old, what people had done was they treated the myosin with proteases that can cleave proteins into chunks. And as just one note on how they work, if you think about it, there are certain regions that tend to interact with there's certain functional domains of proteins, and very often these proteases, they get access to the linkers between these regions, so you can treat a protein with a protease and then do some biochemistry to separate, so to fractionate things on various sizes, and then you can purify these proteins and you can throw them in solution. And one of the experiments that really helped to show that the head of myosin, this region here, has the essential motor function, was to essentially cut off that section of the protein. Now today, most people would just express that section of the protein, throw it down on a cover slip, add some actin filaments, and see if just this head domain can cause motion or not. And this is the result of the experiment. So these are actin filaments that have been labeled up fluorescently. The S1 motor domain of myosin has been attached to the cover slip, and this is what's called photoactivatable GFP. I'm sorry, photoactivatable ATP. And the sort of cool thing here is, well, actually, we'll let the experiment run. So before ATP is uncaged, what's going on? Not much of anything. And then as soon as that ATP is uncaged, you see this dramatic sort of sliding here. So there's two levels to this. One, this really demonstrates that the head domain is capable of sliding actin filaments around. And it also brings up this idea that you can create chemicals that you can manipulate using light. And just to kind of explain, very often what people do is they'll add different sort of atoms to these chemicals, and they're very commonly made sensitive to UV light. So when UV light shines on the molecule, it changes its molecular conformation, and then it allows something that's not functional to become functional. So this is what this uncaged ATP is all about. So you just add the drug, flash in a light of a certain wavelength, it activates the drug, and you can see, what is the immediate effect of having this drug and stuff like that. So I'll just pause for a second and ask if there's any questions on this.
So, obviously the actin filament are moving, but if the head of the myosin itself isn't stabilized by something, how is the filament?
Oh, that's a good question. So the filament, the heads are just stuck down to the cover slip. So a lot of times what happens is you start off and you add something really sticky to the cover slip, and then you throw your protein on there, and it binds, and then you add a blocking agent that's going to, like BSA, bovine serum albumin, is very commonly used. it then gets added, and then that blocks up all of the rest of the sticky sites. And then once that's prepped, you throw them. But I will say, isn't it kind of remarkable? You don't need like a full myosin molecule to slide actin filaments around. So you just need this neck domain. So we've got the essential and the regulatory light chains, the neck and the head. So what that's saying is that's the region that generates that force. So,
Yeah.
okay. And when we look at the 3D models of this, and I almost hesitate to ask, what sort of molecule, DNA, RNA, protein?
Protein.
It's a protein, and we can tell from these ribbon diagrams. So as just a kind of a couple of features here, so we've got the regulatory chain here, the essential light chain, and this is being bound along this alpha helix. This is the head domain, so this would be the neck over here. If you look inside, you're going to see some beta sheets, like there's a cluster of beta sheets. Here's some beta sheets, and up here is where that actin is actually being bound by the myosin. And sort of like with actin filaments, there's a little pocket that ATP can bind. Inside myosin, there's also another pocket that ATP can bind. So, so this itself is the functional motor unit that's generating the forces and stuff. So, okay. So now I want to walk through what is called the cross-bridge cycle, and I'll just pause. Have you guys covered the cross-bridge cycle in class?
Part of the cross-bridge cycle, but just recently I covered like a lot of how this is working.
Okay.
Physiology, we'll get to that yet.
Yeah, so if you haven't had this, it's coming your way. So I find one helpful way to remember the cross-bridge cycle, well, okay, what's one notable thing about being dead?
You don't move.
You don't move a lot. And yes.
Very stiff.
And so there's a phenomenon called rigor mortis. So when people die, muscles get really, really stiff. And how much ATP is being generated when something, yeah, not a lot. So ATP is now, for the most part, absent, and there's going to be a high concentration of ADP. And that, if you keep that one idea in your head, when ADP is present, muscles are locked together. I think that's the place to start with the cross-bridge cycle. So we want to walk through the cross-bridge cycle here. And as just one sort of key idea is that when ADP is present, the myosin heads are going to be locked to the actin filaments, and then ATP is going to come in and release the head from the filament. So in step one, ATP binds, the head is released from the filament, and then the next step involves the extension of that head so it can grab forward. And what's happening here is that ADP is being hydrolyzed to ADP plus inorganic phosphate. And both that phosphate and that ADP are still bound to the myosin. It's just this bridge, this molecular link between the phosphate and the ADP has been broken, but the breakage of that link causes the head to extend outwards. So this enters into the Cox state. Then the myosin can rebind to the filament, and it's as if there's a spring that's been extended and it's fully stretched out. And one thing to think about it, I'll ask, what are the atoms in a phosphate?
Phosphorus, oxygen.
And how many oxygens? So a phosphate is a big molecule. What's the charge on a phosphate? -3. It's a big negative thing. So right now, we've got that myosin head. It's bound to the actin filament. And there's this massive phosphate that's sitting in there. And the next thing that's going to happen is that phosphate is going to pop out. And when that phosphate, you can imagine that phosphate is going to be pushing everything apart. It's going to be keeping the molecule extended. That phosphate pops out, and then a spring can contract. And when that spring contracts, it rotates the head, and it causes a force. And now we're back to ADP, bound to an actin filament. So, okay. So we'll play this and have it in motion. This is flipped around. The phosphate pops out. You see that force. ADP is being released. ATP comes in. It can release. It gets hydrolyzed. It extends, rebinds. That phosphate came out, and then the spring can contract. And that's the essence of the cross-bridge cycle and stuff like that. So, questions or thoughts?
Yeah, I mean, in this example, and in like the examples like we've seen with the photos in the past, like or just in these past couple slides, like you see the second myosin head in the background, but it's not really moving. Is that just because we're not focusing on it, or is that like does that one just not move?
So there's going to be another myosin head, and it's going to be working together with that. And we're going to talk about these, how these heads work together in a bit. But yeah, so
We're just not focusing on it.
We've got to build this up before we can get to that. Yes. Yeah. So the cross-bridge cycle, and certainly a lot of thought has gone into it. But the big idea is that the hydrolysis of ATP generates the energy that is that produces the force in muscles. That's a really abstract way of talking about it. In a sense, I mean, what we have just gone through is how things work. All right. So one thing that is kind of interesting. So this is showing the configuration of the myosin. And one question is how far myosins should step. Do they step? And we can kind of talk about this, So here are individual G-actins. Are they stepping just one G-actin at a time, or is it like 5? How big are these steps? What controls the step size? And what you might imagine is this neck domain here might be important for that. And very much a prediction of this overall sort of model is that step size is going to be a function of that neck domain or the lever arm domain. And a question is, how can we test that? So we've got kind of this working model that the step size of myosin is going to be a function of the length of this neck domain. How would you test that? Go ahead.
You can check the difference in extension of the length of the neck between every 2 steps and see how that length stretches over the actin. Yeah, something like a calculation of.
Yeah. So you can definitely do modeling, and you can look at the length of the neck and the step size. You would need to measure the step size, of course. So, I'm sorry. I thought you had a question. So another way is to just go in and manipulate the system. So if you think something is important, you want to go in and change it and see if it changes the outcome of other things that you think are important. So, and the way that this was actually tested is people created constructs where the length of the neck domain was progressively longer, and then they just measured the velocity of actin filament sliding. And what they found is that the longer the sort of neck domain, this lever arm, the faster the actin filaments slide. And the big sort of question next is, well, how would you go in and measure step size directly? So people have been very interested in trying to understand how myosin generates forces and the size of the steps it takes, the biochemistry of it all. And a really useful tool for measuring very small forces, forces generated by individual molecules, is using what's called a laser tweezer. And in some ways, it's like a tractor beam from like Star Trek or something. One literally uses light to grab a bead, and with that light, you can move a bead around, you can lift it up and down, you can do really just about anything you want with it. And I've used laser tweezers before. They're really, really cool. So kind of the idea is that you'll have like a little glass bead, or you can have a plastic bead, and you want to move that around. And you've got this laser here, it's generating this field of bright, coherent light. You shine it through a microscope objective, and then that light is going to be focused to a little point. And it turns out that that focal point is also the optical trap. And we'll talk about that, how it works in just a second. All right. So, in terms of light, what we have here is laser light coming from this side, and we've got a glass bead here in the center. And a very sort of interesting thing is that whenever light changes direction, it can impart momentum on a system. So you might not think about it, but well, actually, it's pretty wild. People have like used these ideas of solar sails. So you can just take a very large reflective surface, hook it up to a spaceship, and then the photons of light actually bounce off of that and push that sail forward. So light itself can generate forces. And what happens is when light gets bent, there's a small force that's being generated. So if we have a bead that's sitting at the focal point of a microscope and light's coming in, what this is sort of trying to show you here, do you see this curve here in red that is kind of a Gaussian with the peak in the center? So that's representing the density of photons. And what it's just saying is that the spot's right in the center and it's kind of dim on the sides. That's kind of the key idea. And then we have a bead here, and we're showing two sort of light paths here. So in one case, what's happening, so light's entering here and there's a lot of photons, so it's going to go and it bends, and then it's going to bend again, and then it's going to go that way. And we see that the light is being deflected to the right there, and that means it's going to push the beam to the left. There's another group of photons lower in density, fewer in number, on this side, and they're just doing the opposite. So they're trying to push the beam to the right. And because the trap is brighter in the center than on the edges, there's going to be a net force that's going to push the bead to the center of the trap. And if the bead gets moved out of the trap, then that is going to create another imbalance that's going to push it back in. So, go ahead.
So, like by having the bead get hit by two different sources of light, it basically just self-stabilizes.
Exactly, and it's not just two different sources of light. It's more that there's this gradient of light. So this difference in light creates a difference in force, which leads to motion. And it also turns out that because when you focus things, they get really bright along the axis of focus. You can also lift beads up and down. So you can move beads in all 3 dimensions and stuff with laser tweezers. It's really nice. So, okay. But yeah, so this is the sort of fundamental, like the physics of laser tweezers. And one can then drag the beads around and look at how far they move outside of the trap to figure out how strong the trap is. So if you move an object through a fluid, there's going to be a resistant drag force. And based on the viscosity and velocity, you can calculate drag forces for objects. And you can use that to calibrate these tweezers to very precisely measure molecular forces. So these are the optical tweezers, laser traps, and yeah, okay. So, in order to measure the forces and the step size of these myosin heads, people took this even a step further, and they set up an optical trap with 2 different focal points. So they could grab 2 different beads and then use that to grab actin filaments and just move them around. And then they can also take other beads coated with myosin and just get it all set up. So essentially they have this actin filament string with these 2 sort of force sensors on the side in a sense, and then the motor. And we want to look at the step size of these motors. So, to make some predictions, to set up a really simple sort of experiment, so here's the myosin 2 that we talked about, and then here's the myosin 5 that very often transport cargoes. And we can see these neck domains are really different just based on the structure of these 2 different proteins. Would you expect the step sizes, the steps they take to be the same or different? So that's going to be the prediction going in. And these are the outputs from these laser tweezer experiments here. So we have time on this axis, and then we have distance moved in nanometers. And as just to recall, actin is about 5 nanometers. So the jiggle in this line is the displacement of the bead from the center of the trap. And from this, you can see that there's these large peaks that are transient. So it moves and then it gets released, and it's about 20 nanometers. So we want to remember something on step size between 5 to 15 nanometers. So at the extreme, it can be about 20, but there's these smaller steps in here as well. So, okay. So, and I guess one other thing. You just see it like it doesn't go anywhere. In contrast, this is the data for myosin 2. So we just showed myosin 2. This is the data for myosin 5. Here we have time on this axis, and we have distance here, and the behavior is a little bit different. Here we can see that instead of the sort of myosin taking a step and releasing and things popping back to where they were, things are moving over time. In this case, this bead is really moving the distance and stuff like that. So here we can see that the step size is about 36 nanometers. And the first question is, so you look at this and, so that's going to be 36 nanometers. If we go back here, 36 nanometers would be almost to the top here. So is the step size for myosin 5 longer than myosin 2? So that's clear enough. And then the other big difference is that this is what's called a progressive motor, and I'll talk about that in just a little bit, but yes. So step size and lever arm length are correlated. And then the second thing is that the behavior of the beads and the traps are really different. And myosin 5 sticks to these filaments and it keeps moving things along. Myosin 2 just takes a quick little step and then it releases things. And it turns out that in terms of its function, these two molecules, these properties are really important. Mainly because myosin 5 needs to move cargoes, so when it makes forward progress, it wants to stay put and then continue making forward progress. If you think about myosin 2, it's usually setting, like in a muscle or in a cell, it's making forward progress, but it's got to work with a lot of other molecules to do that. And this lack of processivity is one of the things that allows a lot of myosin heads to work together. And I'll have to get to that in just a little bit here, but that's where we're going. Okay, so how do these two arms of the myosin act together? And this is where we get to step size and these ideas of labeling parts of the molecule and seeing how they move and stuff, questions that we had at the beginning. So there's a couple of different models that are used to talk about myosin step size. So one is that myosin can just simply walk. So like as we would walk across here, you can imagine there's other models, and I don't know if my legs are going to show up. I guess not. So another sort of model is that it could do sort of like a shuffle step. Like there's always one like head that reaches out, or there could be other crazy sorts of combinations. It could take half steps or something like that. So we were in those first experiments, the ones that we were just looking at with laser tweezers, we were looking at just single like head neck regions. This is the whole system together. So as I sort of mentioned, so there's a hand over hand model. So you can imagine, and actually this is another way to show it. So the myosin is sort of like going like that, back and forth. And then there's also kind of this inchworm model where it's like it slides and then it can move forward, slides, move forward. So those were two original models. And they make very different predictions about how far things are going to move out of step. And you know, this is one of those things that you just don't know the results until you just look. And it turns out that if you look at the full myosin 5, everything working together, you see the step size is 72 nanometers. And then coming back to this cartoon, oh, I'm sorry. What does that better, what do these data better support as a model for motility here? The hand over hand. And that is how these motors generate forces. So I think this will be a good place to stop here. So our major goals were to explain a little bit more about actin dynamics, but really to focus in on how myosin works and generate forces. So I hope you enjoy.